Hello everyone, I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.G. Hariharan. So today we are going to discuss about one of the important technique of isolation of pure culture, that is streak plate technique. So in this lecture first we understand about what is meant by a pure culture, how the isolation of pure culture is performed, and in the second stage we will elaborately understand how the streak plate technique is performed. So if you see, naturally the microorganism has grown in a complex environment as a mixed culture. That means the different species of microorganism will live together. That means different species of bacteria, different species of fungi will be seen in a mixed form in this natural habitat. So if you want to study a particular types of microorganism, it is not easily done by studying through a mixed culture stage. So first we have to isolate into a pure culture form, then only we can characterize or study that particular species of microorganism. So there is a need for the development of this pure culture. So this technique was actually developed by the great German bacteriologist Robert Koch. Because Koch was developing a particular postulate called as Koch postulates. So in this thing, what he has to prove is that a particular infectious disease is caused by a particular microorganism. To show that this particular microorganism is causing this particular disease means he has to isolate this microorganism in a pure culture form. That's why this pure culture isolation technique was developed by Robert Koch. So the pure culture, what it actually means that a population of cell arising from a single cell of microorganism we regard as a pure culture. That means each cell divides and forms a colony, and this colony will be regarded as a pure culture one. So this pure culture is essential for the characterization of the microorganism of a particular species. So we have to perform various type of morphological characterization or biochemical characterization or even a genetic characterization. We need the organism in pure culture stage. How this can be done? So if you see the basic ideology, how we can isolate a particular species in a pure culture form is that first we will take the sample of the mixed culture, then it is sequentially diluted until the microbial population density is drastically reduced. Then it is spread as an individual cells on an agar surface. Then this agar surface of each individual cells is eventually grow into a colony. And this colony we can see macroscopically on a petri dish. Then it is subculture and used for this various studies of that particular species. So there are different methods isolation available for the isolation of pure culture. The most common methods of isolation of pure culture is that pour plate technique, spread plate technique, and streak plate technique. So in this lecture we elaborately understand about how the streak plate technique is performed. So the prerequisite of starting this technique is that we need a petri disk, a nutrient agar petri disk, which is divided into 4 zones. So in the first step, what we are doing is we are transferring a mixed culture, a loop, sterile loop full of culture to the first sector, either with a loop or a swab. You can add it to the culture. This I have shown in the picture, so you can see that the petri disk, the loop full of culture is added. So in the second step, what it has been performed is the streaking of the first sector. So we take a sterile loop, inoculating loop. This inoculating loop can be sterilized by showing into the outer blue flame of the Bunsen burner, so it becomes red hot and allowed to cool for 10 to 15 seconds. Then the mixed culture is touched, then it is streaked in the first sector. This you can see in the picture. So with the help of the inoculating loop, it is touched and streaked in the first sector. Then the third step is the streaking in the second sector. What happened is that the petri disk is slightly rotated. Then the streaked loop of the first sector is once again sterilized by showing into the outer blue flame of the Bunsen burner. Then it is become red hot. Then it is touched to the first sector endpoint to get the inoculum, then streaked on the second sector. This you can see in this picture. So the second sector has been streaked. So in the step 4, streaking of the third sector. So similarly, the inoculating loop is sterilized once again. The petri disk is slightly rotated. Then the endpoint of the second sector is touched. We get a loop full of inoculum. Then it is streaked in the third sector. This you can see in this picture. So we have streaked in the third sector. Then the final step, streaking in the final sector. So without sterilizing, so the loop carries only few cells of the microorganism. Then the petri disk is rotated. Then it is streaked into the final sector. So this will develop into an individual colonies if it is incubated. This you can see in the picture. The final sector is streaked. So this is incubated, and the individual colonies only we can see in the end of third sector or in the fourth sector. So once again, I will repeat how the steps of this streak plate technique is performed. First, transfer a loop full of organism of the mixed culture into the first sector. Then with the help of a sterile loop, streak the first sector. Then sterilize the inoculating loop and touch the endpoint of the first sector for getting inoculum for streaking in the second sector. Then streak the second sector. Then once again sterilize the inoculating loop. Then touch the second sector. Then streak the third sector. Then without sterilizing, streak the fourth sector. So we will get an individualized colonies once incubated in the end of third sector or in the beginning of the fourth sector. So in the step 4, what we are doing is we are performing the incubation process. The petri disk is closed, inverted, and it is incubated at an appropriate temperature and duration. So after that, the individual colonies will grow in the petri disk in the fourth sector. Then finally it is subcultured by means of transferring the fourth sector individual colonies with the help of a sterile inoculating loop and needle into the fresh medium. Then it is subcultured, and once through we get a pure culture of that particular microorganism, and we can use it for the further studies. So I thank you very much, so that this lecture will be very helpful how to perform on a streak plate technique. So thank you very much. So continually listen my future lectures also.